Swayam Prabha. Digital India. Educated India.
Hi everyone, again welcome to the course of structural biology. We are continuing with the protein module, and today I would teach you about supersecondary structures, motif, domains, their differences, non-covalent interactions which play critical role in protein folding. To start with, before going to supersecondary structures, I want to remind you about something which people are forgetting when we are in artificial intelligence. What is artificial intelligence? We all know it is the simulation of human intelligence processes by machines, especially computer systems. Specific application of AI include expert system, natural language processing, speech recognition and machine learning. AI programming focuses on three cognitive skills: learning, reasoning and self-correction. Whereas, in current era we have seen a lot of progress in the area of machine learning, in the area of artificial intelligence, and that needs technical advancement, that needs appreciation, and they are really doing good. But being in awe with this newly invented technology, somehow we are forgetting about what we call natural intelligence. So, as I told, being overwhelmed with a new generation innovation, which is no doubt wonderful, which is no doubt needs appreciation, we start forgetting the immense power of Mother Nature, her creativity and her ability to control and execute the entire universe. We forget how the entire world is going perfectly, how every day we start, every organism have their own cycle, and how smoothly it is going, and how the overall systems are taken care by the creativity of Mother Nature. Just to show you how amazing the innovative or the creative management of Mother Nature is, I want to put one question. If there are 20 amino acids, as you know already from our course, there are 20 amino acids and they can be assembled in any order, then how many different proteins of 300 amino acid long, which is the average size of a protein, can theoretically be produced? So, to answer it, if we were to build a 300 amino acid peptide, what we have to do? The first position would always be methionine. So, in that position it is fixed that it would be methionine. Other 299 position there would be anything. So, if we were to build a 300 amino acid peptide without much consideration, we would include peptides that would consist, for instance, only of 299 residues, because every protein synthesized by ribosome starts with methionine. There is just one option for the first amino acid, as I told. So, you have 20 and you have to fill up 299. So, the answer is the number of proteins possible theoretically is 20 to the power 299. So, the rest could strictly theoretically be one of the 20. Using the multiplication rule, we would have to multiply the number of possible amino acids in each position by each next number of possible amino acid in each position. There are 299 remaining position. So, the theoretical number of combination would be 20 to the power 299. Now, how big is 20 to the power 299 is? You will be surprised to see that the number is that big. So, basically you could make that many proteins, see the number, that many proteins theoretically. But this numbers of protein does not generate in reality. Why? That is what we call natural intelligence, the creativity, the smartness nature possesses to optimize things. So, one reason which we are going to discuss in the next class is the folding possibilities. Okay? But there are other reasons, and those shows, as I told, the efficiency of NI. What is NI? Natural intelligence. And in this context, I would introduce about super secondary structure. Why? Because nature does not always want significant changes in protein structures, especially in the clusters which are used for critical function. So, think about the function of nature. So many organisms, every organism have their own way, and every way needs proteins. So many proteins are needed, and nature have to develop the rule of their folding. But nature, being creative, being smart, being a wonderful manager, try to find out the commonality. So, Mother Nature have created some clusters, some chain of amino acids, some specific folds which are determined or destined to do some specific function. So, therefore, appearance of signature motif in protein structure is quite frequent for a particular function or a group of related function. How wonderful! Taking an example, helix turn helix is a very common motif, and you see that in MIP protein, in Luxor protein, in ETS1 protein, in MARR protein, everywhere you could see the presence of helix turn helix. So, that is what nature's intelligence is. She is putting one motif in many proteins which are structurally divergent, but have one function common, and that is called super secondary structure or motif. So, what are super secondary structures? They are combination of secondary structures in specific geometric arrangement. What I mean? You have alpha helix, you have beta sheet. So, alpha alpha, beta beta, alpha beta, this type of permutation and combination develops super secondary structures. Super secondary structures are the building blocks of complex super secondary structures and tertiary structures or domain folds. Simple super secondary structures are relatively stable components of the molten globule of protein folding intermediate. Again, we will discuss about molten globule. These are very important part in studying protein folding. So, if you see, we have alpha helices and beta sheets, we talk about them already in details. Beta sheets have parallel and anti-parallel beta sheets. From alpha it comes alpha alpha, from beta it comes beta beta, and from alpha beta it comes beta alpha beta. Then from alpha alpha it comes alpha 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 alpha, from beta alpha beta it comes beta beta beta, and beta alpha beta, and then from beta beta it comes beta 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 beta. The example of alpha 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 are 4 helix bundle, beta alpha beta is beta Minder, beta 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 is Rossmann fold, and beta 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 is Greek key motif. So, super secondary structure are usually produced by packing side chains from adjacent secondary structural element close to each other, and as it wrote, it is between the secondary and tertiary structure. Domain and motifs. Domain and motifs are one of the most confusing terms to separate. So, motifs or folds are particularly stable arrangement of several elements of the secondary structure. Domain, a domain is an independent unit usually stable by itself. It can comprise the whole protein or a part of the protein. When you listen to them, it seems they are kind of same, but there are differences. The key difference could not be understood by doing theory. You have to perform experiment. That is the best way to separate between the domains and motifs. What is the experiment? If you have a protein domain and a motif, you design them, synthesize them separately and try to produce that protein, purify that protein. If you could be able to get the protein produced and purified, then it is a domain, otherwise it is a motif. So, as I told, domains or motifs talk about specific functions, but domains are having their independent existence. If you go to think about evolutionary theory, you understand what is a domain. There are evidences where it was understood or found in the literature that if there is a number of protein, let us say protein A, B, C in a prokaryotic system, in the course of evolution they come into more complicated eukaryotic organism where the function have to be done with much more faster speed, because in complex organism there are more number of proteins, hierarchy of the function and all these things. So, now these three protein, due to implement the work further, comes under one protein by connected with a loops. So, suppose you have a protein A, suppose you have a protein B and you have a protein C in prokaryotes. In the evolution, they comes under one protein. In this situation, each of them are called domains. Motifs are part of the protein or part of the domains, but they do not have ability to work independently. If you cut the loop and try to express, you get to express A as a independent protein. So, that is what domain is all about. Rules for secondary structure development towards motif: hydrophobic side groups must be buried inside the folds. Therefore, if you do that, there would be layers. So, beta alpha, beta, alpha alpha layers are created. Alpha helix and beta sheets, if occur together, are found in different structural layers. Adjacent, because you know alpha helices, they are independently hydrogen bonded together and beta strands do with other beta strands. Adjacent polypeptide segments are stacked together and the beta sheets are more stable. Why beta sheets are more stable? As we have told many times, when you talk about a alpha helix, they have hydrogen bond into N plus 4, whereas and because of that they are actually more stable as a independent unit. Unfortunately, beta strands cannot stay independently, so they are together. Together they have hydrogen bonding if they are parallel or anti-parallel. So, when they are together, they brings more stability, more energy to the system. So, that is why beta sheets are more stable. Coming to the motif, as I already talked, secondary structural composition, all alpha, all beta, segregated alpha plus beta, mixed alpha beta. Motif is small specific combination of secondary structure elements connected by loops. Constructive large motifs from smaller ones, the alpha beta barrel is a commonly occurring motif constructed from repetition of beta alpha beta loop motif. If you see, this is the beta alpha beta loop motif. This is present here in the bigger protein. So, this beta alpha protein is part of the pyruvate kinase, which is a glycolytic enzyme from rabbit, where you see that with a PDB ID 1 PKN. If you remember, in our first module, the introductory module, I have introduced PDB ID and UniProt ID. PDB IDs are common for a protein's unique structures, like one PDB ID is unique for one structure, but one protein could have many PDB IDs, whereas one protein would have only one unique UniProt ID. Coming to different motifs and their example, beta hairpin is a very known motif. So, beta hairpins are one of the simplest supersecondary structures and are widespread, especially in globular proteins. The beta hairpin is a simple protein structural motif involving two beta strands that look like a hairpin. If you see here, one beta strand, two beta strand connected with a loop, and it look like a hairpin. The motif consists of two strands that are adjacent in primary structure, oriented in an anti-parallel direction. So, because of that, it goes in an anti-parallel direction. The end terminus of one sheet is adjacent to the C terminus of the next and linked by a short loop of 2 to 5 amino acids. So, depending on how many amino acids develop loop, they have their classifications. So, you see 2 residue beta hairpin turn example here, 3 residue beta hairpin, these are the examples. Beta hairpins can occur in isolation or as a part of a series of hydrogen bonded strands that collectively comprise a beta sheet. Here you see in isolation, here you see it comes in a series. Example of beta hairpins presence in protein, this is tryptophan zipper, this is having the beta hairpin you could see. This is the example of beta hairpin in bovine pancreatic trypsin inhibitor BPTI. BPTI fold is a very classic, stable and frequently found fold in nature. Example of a protein with 2 beta hairpins, arabotoxin from whale. Beta hairpin have also very interesting aspect that their presence are there in many toxin proteins. Coming to another one with longer loops is called beta mander. For this, a wide range of conformation is observed and the general term random coil is sometimes used. Consecutive anti-parallel beta strand when linked by hairpins from a super secondary structure known as beta mander. If you see here, there are anti-parallel beta strands and they are connected by loops. And when I am talking about loops, I want to talk about different turns and loops. Beta turn we have already talked about. Donor and acceptor residues of hydrogen bonds are separated by 3 residues. So, I2 I plus 3 hydrogen bonding, but there are many other turns like delta turn which is I2 I plus 1 hydrogen bonding, gamma turn which is I2 I plus 2 hydrogen bonding, alpha turn which is I2 I plus 4 hydrogen bonding, pi turn which is I2 I plus 5 hydrogen bonding, and omega loop, a longer loop with no internal hydrogen bonding. So, it kind of look like omega, so it is called omega loop. Omega loop is present in many protein, and when present they take very interesting role in catalysis. They influence the active site volume, they interact with the substrate. One of the very popular example of omega loop is class A, mostly it is found in other subclasses of beta lactamase, but beta lactamase, especially in class A beta lactamase, they are very intensely studied, and it is found that they have very critical role in the catalysis of the enzyme beta lactamase. Coming to other one which is called beta corner, anti-parallel beta strand forming a beta hairpin can accommodate a 90-degree change in direction known as beta corner. If you see, there is a 90-degree change in the direction. The strand on the inside of the bend often has a glycine at this position, so that the flexibility comes, while the other strand can have a beta bulge, the extension. The latter involves a single residue in the right handed alpha helical conformation, which breaks the hydrogen bonding pattern in the beta sheet and help it to turn. This residue can also be in the left handed helical or bridging regions of the Ramachandran plot, as we have discussed in our last class. Beta corners are observed to have a right handed twist when viewed from the concave side. This is an example of a beta corner. Beta alpha beta motif. So, up to now the motifs we talked about concerning beta strands are mostly in anti-parallel, but beta alpha beta motif is where parallel beta strands are connected by longer region of chain which cross the beta sheet and frequently contain alpha helical segment. So, it is beta beta in between alpha. The motif is called the beta alpha beta motif and is found in most proteins that have a parallel beta sheet. The loop regions linking the strands to the helical segments can vary greatly in length. The helix axis is roughly parallel with the beta strands, and all three elements of secondary structure interact, forming a hydrophobic core. The beta alpha beta motif almost always has a right handed fold. The helix turn helix motif, which we have started our discussion of supersecondary structures with, it is a very popular one. It is characteristic of protein binding to the major DNA group. So, if you know about DNA, in DNA there is major group and minor group. So, helix turn helix, it goes there and bind to the major group. The proteins containing this motif recognize palindromic DNA sequence. What is palindromic? Palindromic is, if I write the word madam, so if you read it from this side, it is MADAM, madam, same. From this side, madam. So, if you have a word which could read from both the side coming up with the same thing, it is called palindromic. So, what or how it is connected with the DNA sequence? Similarly, in DNA sequence you start reading from both the side, you see or you get same sequence. The second helix is responsible for nucleotide sequence recognition. So, the first alpha helix goes and bind to the major group, and the second helix is responsible for nucleotide sequence recognition. So, you see the binding in the helix turn helix motif. Very interestingly, here you have two helix turn helix motif. One is homeodomain, which is coming from eukaryote, and one is lambda repressor, which is in prokaryote. So, it clearly says that if you compare, the prokaryotic protein is having helices with smaller chain length. So, here if you see this chain length here is much bigger than here. So, that says that in evolution from prokaryotic to eukaryotic there is a change. The change is the size of the helices are significantly larger in higher eukaryote. Many DNA binding proteins are dimer. For example, most of the DNA binding protein which belongs to TCS. What is TCS? Two component system family. Two component system is a system which we will discuss later. It is a very interesting system which help bacteria to survive in its critical moment. In the TCS system, there is one histidine kinase, component 1, and 2 response regulator or is called aspartate kinase. So, they bind by making dimers. This means that there are two helix turn helix motifs per dimeric protein, one coming from monomer A, other coming from monomer B. This will interact with two adjacent major groups, that is 10 base pair apart, and nowadays similarities, patterns are used in technology development. So, this is an amazing pattern. Here you see that you see the DNA per turn there are 10 residues. So, if one part or one atomic interaction happens at 12th residue, the second one from the other dimer would happen after 10 base pair, the same interaction. So, that is very interesting, and the DNA recognition site is therefore frequently an inverted repeat. If you look at the DNA binding closer, you will see that specific amino acid on the side of the helix facing DNA interact with the base pair edges through hydrogen bonding. So, there is phosphate, phosphates are taking part in interaction, but the nucleosides are also. The number of interactions between helix 2 and the DNA sequence determines the strength of DNA binding. So, the first helix goes to the major group, the second helix is interacting with the nucleotides, and depending on the difference in the nucleotide, the strength of the interaction differ. So, what is the significance? The significance is, I told nature is extremely smart. So, these proteins, these DNA binding proteins tells the organism about a situation. Let us say it is telling the bacteria that somehow where you are staying the heat is increasing, so you have to escape if you want to survive. Okay. Now, heat is not a specific term, it is a relative term. It might be low amount of heat, medium amount of heat, high amount of heat, and even many other things in between. How bacteria could sense by getting the signal that this is very high heat or this is low amount of heat? That is, the difference is done by binding to nucleotides in different proteins. So, it comes and it bind to specific site, get interaction. So, let us say it binds to site A, B and C. Here the interaction is highest, here it is medium and here it is low. It is the same protein, but binding to this side it would provide a message that very high heat is coming. Here medium heat is coming, here low amount of heat is coming. Why it is very critical? Depending on them, this will produce the number or amount of heat shock proteins which will help bacteria surviving, but bacteria also do not want to do more work than needed. So, when it is low, it would produce less amount of protein in that way. So, this explain why the same protein can bind to different yet related sequences with different affinity, giving different signals. So, see how again nature plays smart, one protein, but different sites in the DNA. Which side it is binding determines or says about, provide the signal what is the condition. Coming to another one, alpha alpha corner, short loop region connecting helices which are roughly perpendicular to one another are referred to as alpha alpha corner. You see the picture of alpha alpha corner, two alpha helices connected by a loop. The C terminal residue of the first helix must be a short side chain to avoid steric hindrance and is observed commonly to be glycine. As you already know, glycine is the smallest. The first residue of the second helix, which is in the beta conformation, frequently has a small polar side chain such as serine or aspartate, which can form hydrogen bond with the free NH group at the amino terminal end of the second helix. The central residue of the alpha alpha corner is almost always hydrophobic, as it is buried and interact with other non-polar side chains buried where the ends of the two helices contact each other. So, I discussed about different motifs. There are so many motifs and they have very interesting and critical roles in the signaling pathways. To understand biology, if you understand the super secondary structures, it would be extremely helpful. Zinc finger domain, it is also a DNA binding domain. B zip, another DNA binding domain. I am generally talking about DNA binding domains because DNA binding domains are mostly transcription factors, they work with signal transfer, they are very critical and very well understood mostly. SH2 domain is not DNA binding, EF hand domain, death affected domain, PTB domain, armadillo repeat domain and PH domain. They are some commonly known domains. So, how could you apply the knowledge of domains? While discussing about, I have already introduced the importance, but I want to end up with the thing that super secondary structure or motif or domain have another very critical role. You know, when I introduced the course, one of the biggest, most critical problem I talked about, sequence, we get sequence from NGS, I am repeatedly talking about. We want to get function and we need structure. In the next modules, we will continue with the structural biology technique in details, but in our previous introductory module I have already talked about, it is kind of impossible situation in present scenario to make a huge speed up in structure development techniques, so that we could make the bridge connected. The alternatives, and one of the most accepted alternative is structure prediction. I know when I am talking about this, some of you definitely argue about the artificial intelligence, about AlphaFold, which are impressively solving structure problems now. But if you look at the bigger picture, unfortunately there is many more things to do. So, when we are doing structure prediction, be it any prediction modeling method, simulation, energy minimization, or artificial intelligence, getting the knowledge of domains would be immensely helpful in understanding about a protein. What I mean, let us say you have a big protein. If you know that this part, this supersecondary structure, there is a supersecondary structure and you know the function, this is a fantastic clue towards solving the structure. So, this is one of the very good application of domains. Along with in enzyme engineering, it play critical part. Coming to the next part of the topic today, which is weak forces. Weak forces in biology. Covalent interaction establish the structural framework of protein molecule, the chemical expression of primary structure. Backbone conformation constrained by steric restriction on phi and psi torsion angles. Side chain conformation, which are determined by the chi torsion angle, are also constrained. Favorable side chain conformation depends on the side chain and also on its neighbors. So, it is not only about the amino acid present there, it is also about the neighbor. If you have glycine and glycine, if you have glycine and arginine, if you have arginine and arginine, they will behave very differently. And about disulfide bonds, they are the only covalent bond which could form in physiological condition, they could alter. So, we will discuss about that. Inter and intramolecular force, the covalent bonds hold atoms together so that molecules are formed. Weak forces profoundly influence the structures and behaviors of all biological molecules. Weak forces create interaction that are constantly forming and breaking under physiological condition. The energies are 0.4 to 30 is an approximation, it is around 100 you could say. So, what we are talking about? We are talking about that a protein primary structure we get from covalent bonds, mostly sigma, but you also have pi. Then our journey is from these to secondary, then tertiary, and this ends with 1. If you have more than 1, then quaternary. This part, the journey there, and also the change of conformation, Why change of conformation is very important? Because for protein molecule to be functional, it have to show the change when it want to start a reaction, when you want to finish a reaction, when it want to work, when it do not want to work. All these happen through the change of conformation. So, change of conformation is very important. This needs breaking and making of non-covalent bonds. So, before going into non-covalent, as I told, I will discuss about disulfide bond. A disulfide bond, also called a SS, sulfur sulfur bond or disulfide bridge, is a covalent bond derived from thiol groups. In protein, disulfide bond formed by the oxidation of sulfhydryl group of two cysteine residues. We have only two amino acids, cysteine and methionine, which have sulfur, but methionine having a CH3, a poor Lewis group, it cannot be taken out. So, it could not form disulfide. Cysteine is the only amino acid forming disulfide bond. Disulfide linkage formed during the secondary structure construction of protein, and it also plays crucial role in protein's thermal stability. By introduction of one or more disulfide bond, you could increase considerably thermal stability of a protein. So, you see when two cysteine, they are in close proximity in the protein structure, in oxidizing condition they form disulfide bond. Application of disulfide bond: to increase the thermal stability of protein, we can introduce favorable disulfide bonds. So, yes, when we compare between two proteins, the presence of disulfide bond differs, but this gives us idea about engineering. When you have a protein, you want to make it thermostable, you could have approximated or strategized introduction of two sulfur or two cysteine, so that they could form a disulfide bond. For this purpose, we have to identify the region where we can introduce a disulfide bond without any issue of steric classes. For introducing a disulfide bond, we have to mutate two residues with cysteine, maintaining proper geometry of forming this type of covalent linkage. In this case, we can take help of concept of finding nearest neighbor to investigate the total protein. We will talk about finding nearest neighbor concept when we will discuss the enzyme engineering or protein engineering. So, when you have to do those calculation, you have to understand the geometry very well. So, I have given the details of two cysteine collected from many proteins who are successfully forming disulfide bond. This information being into your database would help you to design a new cysteine pair in a protein, and we will discuss about that in details. So, weak force include ionic interaction, hydrogen bond, van der Waals interaction and hydrophobic interactions. There are base stacking and all these things, but mostly these four are taken care. Ionic interaction is real charge interaction. The presence of real charge makes it happen in energy close to covalent bond. Hydrogen bond, as approximated 2 to 20, it differs depending on the situation, but it is a low energy. Van der Waals interaction 0.4 to 4 kilojoule per mole, and hydrophobic interaction 3 to 10 kilojoule per mole depending on the patches. So, ionic interaction, as I told, charge side chains of amino acid in protein can interact favorably with opposing charge of another side chain according to Coulomb's law, F equal to Q1 Q2 by dr square, where Q1 and Q2 are two charge entity, R is the distance, D is the dielectric constant. The requirement here is the development of genuine charge, like a negative charge in a carboxylate ion or positive charge in a ammonium ion. Salts have the ability to shield the electrostatic interaction. So, charge interaction, it happen in protein as I told, but it could happen between a metal with the phosphate in nucleoside or any phosphorylated protein. So, Coulombic interaction occurs between two ions. In a simple term, at close range Coulomb interactions are as strong as covalent bonds. Their energy decreases with 1 by r. So, with increasing distance energy decreases and fall up to less than kT thermal energy at about 56 nanometer separation between charges. In practice, charge interactions have been shown to be chemically significant up to 15 angstrom in proteins. Small charge metal ions can act as positive charge in an ion pair. So, these are another example of amino acid. So, when this happens, these ionic interactions you see here, we call it salt bridge. So, let us talk about salt bridges. The salt bridge is one of the most important molecular interaction in protein. A salt bridge is actually a combination of two non-covalent interactions, hydrogen bonding and electrostatic interaction. If you see here between glutamic acid and lysine, you see that this is the charge, real charge form, and they are having the electrostatic interaction, but when it is not between the real charge, a hydrogen comes inside, it is called hydrogen bond. It is very difficult to separate them conceptually. It helps to maintain structural integrity of a protein and also holds protein's backbone rigidity at high temperature. Salt bridges form between two oppositely charged residues. Mainly 4 residues of 20 amino acid remain involved in salt bridge formation, which are chemically distinguished into two properties. One is basic, which is arginine and lysine, as others are acidic, aspartate and glutamate. So, geometry of salt bridge is very important. The centroids of the side chain charge group in oppositely charged residues should be less than equal to 4 to 5 angstrom from each other. Pair of aspartate or glutamate side chain carboxyl oxygen atoms and side chain nitrogen atoms of arginine and lysine should be less than equal to 4 angstrom. So, here if you see the green spot, it talks about the centroid. The brown dotted lines are the distance between centroids and the red dotted lines are distance between oppositely charged group. So, these are different example of salt bridge in protein. This is presented in a crystal structure where you see the electron density. This is presented in a by a software COOT, which we will discuss. Then this is a glimpse where one arginine and one aspartate are developing, and this is a salt bridge network. So, this is a single salt bridge, single residue salt bridge. This is salt bridge network. So, you see the differences. Application: using concept of nearest neighbor, which I talk in case of disulfide, we can scan through a protein to identify pair of oppositely charged residues and check if they satisfy the geometry of the salt bridge formation. By the same way, we can also identify the destabilizing salt bridges. So, destabilizing salt bridge is a concept which I really worked on personally. This is an amazing concept of nature. Suppose this is a salt bridge. Because it is oppositely charged, we are telling it as stabilizing salt bridge. Now, a stabilizing salt bridge would help stabilize a protein. So, here you know that for positive charge you have arginine or lysine, and here you have aspartate or glutamate. Now, you see, if you mutate this, let us say to alanine, then the positive effect would be neutralize, they would no more stabilize, right. Think about, instead of doing this, if you model a same charge like a arginine here, let us say, then they will repel each other. So, that is what we say destabilizing salt bridge. Now, if you are listening carefully and understanding the concept, you will say, why nature will do that? You say nature is smartest and by making a destabilizing salt bridge, nature would actually introduce adverse effect in a protein. But if you think what I am saying, suppose you have a thermostable protein, nature want to make it mesophilic protein. So, nature did that neutralizing effect, but if it do the destabilizing salt bridge, then from this thermostable it could make a protein which work in cold condition, that is called psychrophilic protein. So, you understand what I am saying? So, when you have a thermophilic protein, by introducing a destabilizing salt bridge concept, you introduce huge flexibility on that portion, and in course of evolution that might be performed to convert a thermostable protein to a psychrophilic protein. That is where we introduce the concept of destabilizing. By identifying the destabilizing amino acid or destabilizing salt bridge and change it to a stabilizing salt bridge would have double effect towards stabilization. So, this is a very interesting strategy in thermostable enzyme or thermostable protein engineering. The application would draw some idea for the user about the stability of a protein in aspect of containing stabilizing and destabilizing salt bridges. Coming to the next topic, van der Waals forces. Van der Waals forces are also known as London forces. They are weak interaction caused by momentary changes in electron density in a molecule. So, in one word, if you have a molecule and because of its stretching, bending, somehow because of dynamic changes, the electron distribution changes, there is a dipole development, that is where you get the van der Waals. They are the only attractive forces present in non-polar compounds. As an example, even though methane has no net dipole at any one instant, its electron density may not be completely symmetrical, resulting in a temporary dipole. So, as I was discussing about, as this picture is discussing about, so when there is bond stretching and all, there is difference in the electron density distribution, as you see here, unsymmetrical electron density, that creates a dipole. So, you see here a presence of partial negative and partial positive charges. This can introduce a temporary dipole in another molecule. The weak interaction of these temporary dipoles constituents of van der Waals forces. The surface area of a molecule determines the strength of the van der Waals interaction between molecules. The larger the surface area, the larger the attractive force between two molecules and the stronger the intermolecular. So, if you see there is some amount of negative and positive charge developed, now if the area would be doubled, then you have more amount of charge difference. So, how it happened? If you have two atoms coming together, initially the electrons of one attracts the nuclei of other. So, when it starts from this point, because of this attractive force, there is energy reduced. So, this is energy of attraction between the electrons and nuclei. But when it comes to certain distance, then electron nuclei repulsion started, and it would increase in the energy. Okay. this is the minimum energy distance and this is the equilibrium distance. It decreases with the power of r to the power 6, where r is the distance, approximately 1 kilojoule per mole. If r 0, which is the equilibrium distance, is the sum of van der Waals radii of the two atoms, van der Waals forces are attractive forces in this region when r greater than r 0 and repulsive when r less than r 0 in this. This is the repulsive, this is the attractive. Coming to the next one, hydrophobic interaction. The hydrophobic effect is the observed tendency of non-polar substances to aggregate in an aqueous solution and exclude water molecule. So, they are hydrophobic, so they fear of water, they want to exclude the water. The hydrophobic residues glycine, alanine, proline, valine, leucine, isoleucine, methionine are mainly involved in hydrophobic interaction in protein. So, these are the given amino acids, but in addition to that phenylalanine is also there. So, they provide hydrophobicity to the protein molecule. Why do hydrophobic interaction occurs? There has some argue that the hydrophobic interaction is mostly an entropic effect originating from the disruption of highly dynamic hydrogen bonds between molecule of liquid water by the non-polar solute. If you see the leaf of lotus, where if you see that if you put a water, it will create a droplet because of hydrophobic interaction. A hydrocarbon chain or a similar non-polar region is patch of a large molecule is incapable of forming hydrogen bond with water. These molecules are also known as low water soluble molecule or hydrophobes. Hydrophobic interaction play very important role when protein or enzyme interact with the substrate. So, hydrophobic interaction in protein can stabilize any motif or domain conformation. This interaction also can play important role in protein ligand complex formation. If you look at here, the leucine, alanine, leucine, they are interacting with the hydrophobic part of the drug molecule. So, in the structure of Mycobacterium tuberculosis beta-lactamase, the residues alanine 133, leucine 137 and leucine 140 shown to have hydrophobic contribution during the interaction with the compound NXL 104. The green are the carbon part of the hydrophobic part of the compound NXL 104, which is also commonly known as avibactam, a very interesting new generation drug which could have inhibit different beta lactamase enzymes. Hydrophobic cavity formation in protein. So, hydrophobic residues has significant role in the formation of cavity inside the protein structure for the accommodation of any non-polar substrates. Presence of hydrophobic residues allows the cavity to become flexible, so that the residue can change their conformation to occupy the vacated space according to the movement of the substrate molecule. So, here you could see the green patches in the figure, which are hydrophobic cavity in protein structure in which a fatty acid long chains would be nicely packed. So, that is another example of protein ligand interaction by hydrophobicity. Coming to the next one, the last one, hydrogen bond. Hydrogen bond, it is a non-covalent chemical bond in which an electronegative atom, a hydrogen bond acceptor, shares a hydrogen atom with an electronegative atom with bound hydrogen. So, mostly oxygen and nitrogen in biology forms hydrogen bonds. Energy 1 to 40 kilojoule per mole, approximate length 1.7 to 3 angstrom in length. Strength varies with angle of hydrogen bond interaction. Individually not very strong, but the large number of hydrogen bonds in regular secondary structures stabilize the framework of the protein. So, what it is talking about, that hydrogen bond probably plays the most critical role in a protein function. As I talked about, the protein function needs the change of the protein conformation, so it needs constantly breaking and making of bonds. Hydrogen bonds being very less in energy could have formed and could have broken very easily, and by doing that and doing it in a huge number, they help in changing the total conformation of a protein, and that's kind of take role as a switch on switch off in protein function, which is extremely critical. Also, we talked about the strength varies with angle of hydrogen bond formation. Here I want to talk about the two very popular motifs, the alpha helix and beta sheets. If you see, we already talked about the hydrogen bond between CO and NH is formed. So, we talked about the angle dependence of a hydrogen bond. Distance dependence we understand, but angle dependence is not understood very easily. Here is a very good example where we are taking the classic secondary structure motifs, which are alpha helix and beta sheets. So, if you see, all of them form CONH bond, where in alpha helix it is from N to N plus 4th residue, and it is face to face. Here you get the oxygen and the nitrogen in this fashion. Interestingly, here how you get it, in parallel you get it like angled. So, this is parallel beta sheets. In anti-parallel you get it face to face. So, individually by calculating the hydrogen bond, the most stable alpha helix, second anti-parallel beta sheets, and third parallel beta sheets. So, stability wise this is the order, but as I talked that beta sheets cannot be staying on the, so I should write it as strands. But as you know, how you form a parallel or anti-parallel strand, they are already sheet, and that is why they have higher energy. So, in terms of energy it is misleading. Individually alpha helix, because of its proper angle and distance, form the most stable unit, whereas beta strands are coming together and it is not possible to measure their energy individually, because individually there is no hydrogen bond. So, we have to measure the entire sheet, and that is why they show more energy. So, we are done with this discussion. There are so many non-covalent bonds, we know about them, but the interesting part is that how they come together. We have studied their individual effects and their physics, chemistry which are according to them, but it would be really interesting how they work together, and that gives rise to a phenomena which is called protein folding. As you see from the history I have talked about, protein is studied for more than 100 years, but and one of the focus of studying protein is to understand its folding. if we could understand protein folding, we would be close to immortality. So, protein folding is still an enigma, and in spite of the efforts of many biochemists, biophysicists, structural biologists. So, in the next class we will talk about how protein folding happen, what are the thermodynamic phenomena associated with them, and more importantly how we could study those things. Thank you very much.